Okay, part 2 video focusing on DNA replication. This is where I didn't really like the way it was going the first time around, and so I broke it up just in case this doesn't go as planned. DNA replication is a tricky process, so you really have to pay attention. You really have to understand that whole idea of 5 and 3 prime, where it does become overwhelming. First thing, DNA is a double strand that has been connected by hydrogen bonds. So the first thing we have to do is actually separate those DNA strands using a new enzyme, helicase. I know you probably can't see this detail, but this is what's in your notes, so I don't technically have to do so much with this. To show you what helicase does, I'm going to draw my DNA molecule very, very simply. I don't even have to draw the nucleotides yet, because that's, once again, not truly the focus of this. If this is an example of what DNA could look like, it's the backbone, and then these rungs are like the 2 nitrogen bases that are connected. Helicase acts like a zipper. If this is a closed DNA, it's like a closed zipper. The helicase acts as technically the zipper that comes in, and it starts to unwind the DNA strands because it separates the nitrogen base from the top strand from the nitrogen base from the bottom. Recall that nitrogen bases are connected by hydrogen bonds. They're really weak. They're just attractions technically. So an enzyme can go in and start to break these up. If DNA was combined because of covalent bonds, it'd be a lot harder to break because covalent bonds are a stronger bond. But these hydrogen bonds, they're a little weaker, so therefore it can separate a lot easier. So here's my little, it's like a wedge that goes in and it breaks it up. So this is before, and then I'll show you what it can look like in the middle. So I could still have some connected, but what will start to happen is the DNA, these strands have been separated because there's my wedge. It's kind of like in the middle of the DNA. So some strand is still double connected, and then these strands are single. And that's all helicase's job is it goes down the strand of DNA and it separates it. In the next model, you see that the 5 and 3 primes have been identified, so that's what I will do. I'm going to keep it consistent. They're saying that this end up here is the 3 prime end, so mark your notes there as 3 prime. There's the 5 prime, so therefore on the opposite side, there has to be the 3 prime, has to be the 5 prime. Once they have been exposed, these are single nucleotides. What your body has the ability to do now is it starts to add single nucleotides. Your nucleus just doesn't have all of these double strands. It actually has free-floating nucleotides in it that are ready to come in and start to connect and make new strands of DNA. Now, why do we have to do this? The whole purpose of DNA replication is the idea that when you grow as a living character, like a living body, your cells don't get bigger. What they do is they go from 1 cell to 2 cells, and every cell has to have a nucleus, so every cell has to have DNA to it. So if you go from 1 cell to 2 cells, this 1 cell has to actually duplicate itself, and so it has to replicate the DNA within so that the original cell has the original DNA, but then you also have a new cell that can get the same exact copy of DNA. Just a little tangent, but you got to understand the big picture if you really want to understand why we're doing this. I don't know where I got this diagram. It's wrong. If we look at it, for some reason they don't pair them up correctly. If you look here, G to C is right, C to G is right, A to T is right, but then here there's like a little mutation. So I want you to change this C, you can change it to an A. If we're keeping consistent, the next one also is a mutation. This should be a C. Then they pick up and say T to A, that's fine, G to C is fine, C to G is fine, and then they have 2 random ones. So we've got to make up for that up here, so we might as well, if this is an A, what does this have to be? T, we'll extend the line, and then this has to be a C, and then it's perfect. Alright, we just fixed it. No reason to complain. Still, this is now the 3 prime end out here. This is the 5 prime end. And then all the way back here, 5 and 3. This strand is now going to get a new complementary strand. Just like this old strand, notice it says old, it's going to get a new complement. But this is where the tricky part comes in. It's not equal on both sides. One strand is very easy to form. The other strand is a lot more difficult. So we have to be able to identify the easy strand versus the hard strand. What I suggest you do first is just put the first nucleotide down opposite the old strand. So if this is the old strand, and I'll change my color to red. If this is a C, we know on the new strand coming in, the first one is going to be a G. And then I'll draw, there is the rest of the nucleotide. So this would be the sugar and the phosphate in a simplified diagram. If this is a G, the new nucleotide that's coming in through replication has to be that C. And then we'll draw the backbone portion of it. So that's not a T, that's the sugar phosphate. My suggestion, this is where we have to identify the 5' and 3' just on one nucleotide. I'm not going to draw a whole strand. You should still be able to do this. If the right side of this cytosine, which is the last one on the old strand, has the 3' end, what does the right side of my guanine, which is on my new strand, have to have as a number? Did you say 5? If you did, you are correct. If you did not, you need to go back to lesson number 1. On the left side of this guanine, remember there's a sugar here. One side has a 5. Guess what the other side has to be? That's my 3'. You don't have to draw that yet because we're actually going to draw more DNA going down. On this cytosine, if the right side of the old strand had the 5' exposed, what does the right side of my new cytosine have exposed? That has to be the 3'. And then my left side of the same nucleotide, remember, even one nucleotide can have a 5' and a 3' end. That's what we're identifying right here. So if this is a 3', this then has to be a 5'. Now, why did I do all that work with the 5's and 3's? It's because this sentence right here, addition of new nucleotides to the exposed single-stranded DNA molecule uses an enzyme. It's the second enzyme we need to know. It's called DNA polymerase. Polymerase, polymer, it's making something larger. We're making a large nucleotide using an enzyme, and it's going to make DNA. There's a rule though. Unfortunately, DNA polymerase is not a very versatile enzyme. It only can go in one direction. It's like a one-way enzyme. If it could work in both directions, DNA replication would be completely different. But it can't. It can only go in one direction, and One Direction. Isn't that like a new boy band? If I knew one of their songs right now, I'm not very good at it. Okay. Are you singing one right now? I think they just have... No, that's Justin Bieber. I'm thinking Baby, Baby, Baby. There you go, Hannah. There's my Justin Bieber reference. So here's what we have. It can only add nucleotides in a 5' to 3' direction. So I want you to put your pencil down on the 5. We're looking at the new strand. There's my 5. There's my 3. That's the direction that DNA polymerase is capable of moving on the new strand. It can go from 5 to 3, which is the same direction as the helicase is moving. That's a good thing. This is going to be the easy strand because the direction of the polymerase is going to work in the same direction as the helicase is working. Here's the wah-wah. This is going to be the hard one to build because look at it. Here's the 5'. It's on my left. Here's my 3', it's on the right of the nucleotide. So the direction of this DNA polymerase can only go 5 to 3. It's going in the opposite direction as the helicase. So that's going to be a lot harder to do. So we're going to have to come up with a way to figure out how to add nucleotide that's going to be completely different than this one. So what I want to do is just show you this top strand. This is the direction which the bottom strand is going to work in. It's called the leading strand. We're going to define it a little bit, but I want to identify right now. Leading strand. It goes from 5 to 3 in the same direction as my helicase. So the next nucleotide that I add could literally be the one that would be across from the second nucleotide on the old strand. So if this is a G, the next one, and that's why I'm going to erase this, but I technically don't need it. Once I've identified the leading, that's the only reason I put that there. So the next one that comes in, I can add as an A. It can add, because it's going 5 to 3. The next one, C. The next one, G. The next one, T. C. A. C. I think that's where I'm going to stop today's lesson because this other one might need a little bit more help in class. So today's lesson at home is to understand the first step, helicase, and understanding the idea of how to identify 5' and 3' on the new strand. I hope that worked. That was much better than I think than my first go at this because I was trying to shove a lot more down than I think that could handle in 15 minutes. So tomorrow in class I will talk about this. It's not going to be a completely flipped lesson. So tomorrow in class we will learn a little bit about this second strand. Until then, stay classy, North Bend.